Coming to the last phase in karyokinesis is the telo phase. Here telo indicates end. Telo indicates end. See pro early, telo here end. Right, prophase, meta, center, right, and a movement of chromosomes, telo end, right. So that is about telophase name, right. Coming to the next one, within telophase, okay, within telophase some changes that takes place. Within telophase some changes that takes place which are opposite to prophase. Opposite to prophase means whatever the changes that takes place in prophase, okay, that changes are exactly opposite in telophase. The changes are exactly opposite in telophase means I like to discuss. It's very easy. If we remember prophase, opposite to the prophase is the telophase. Opposite to the prophase is the telophase. That's the reason why. Okay, this one is the reverse prophase. Reverse prophase. See, within this phase, this is the cell. This is the cell. Okay, we already know the chromosomes are reaches the opposite poles. The chromosomes are reaches the opposite poles. Once the chromosomes are reaches the opposite pole side, there is a decondensation of genetic material. Decondensation of genetic material. See, generally we know the concept is within the cell. Okay, if the chromosomes, whatever the chromosomes are there, which are moving towards the opposite pole side, right? Once these are reaches the pole side, decondensation of chromosomes. Remember, in prophase, condensation of genetic material. In prophase. In telophase, decondensation of genetic material. That is nothing but decondensation of chromosomes. And next point is, okay, condensation of nuclear membrane. Condensation of nuclear membrane is nothing but, I already said, formation of nuclear membrane. Nuclear membrane formation takes place. Right? So that is about the formation of nuclear membrane. In prophase, in late prophase, okay, nuclear membrane is ready to disappear. See, in telophase, the nuclear membrane is appear. Right? So that is about the second point. And condensation of nuclear membrane. And next point is condensation of cell organelles. Means we already discussed, if the cell organelles are present, see, there is no movement of chromosomes. If cell organelles are absent, the chromosomes are easily moving towards the opposite pole side. So for that purpose, whatever that cell organelles are there, all these cell organelles are disappeared in the late prophase. All cell organelles are disappeared. So when the cell organelles are appeared again, within telophase, all cell organelles are appeared. So these are the points which are related to the telophase. Remember, telo indicates end. Right? This is the end phase in karyokinesis process. Karyokinesis is related to mitosis. So here, decondensation of chromosomes. That is nothing but whatever that the chromosomes are there, which forms the chromatin structure. And next one, condensation of nuclear membrane. After the separation of genetic material, after the separation of chromosomes, once which reaches the opposite pole side, immediately nuclear membrane formation takes place. Right? And next one is condensation of cell organelles. Cell organelles are very important. Right? See, energy is required. So within whatever that energy which is synthesized in G1 phase and S phase and G2 phase, mostly energy synthesis takes place in G1 phase and the G2 phase. Right? So that energy utilized because within metaphase, within anaphase, the cell organelles are disappeared. Right? There is no ATP synthesis. So when the ATP synthesis takes place, the ATP synthesized, mostly in G1 phase and the G2 phase. Okay, whatever the energy which is sensitized in G1 phase and the G2 phase, that energy utilized within karyokinesis process. Within karyokinesis process, right? So that is about that cell organelles formation, nuclear membrane formation. I like to discuss about here one of the point. Okay, if the cell generally follows the cytokinesis process, most of the cases, 99% of the cases, okay, most of the cases, if the cell follows the cytokinesis process, in that case, okay, one cell divided into two cells. One cell divided into two cells. So this cell contains one nucleus, this cell contains one nucleus, right? So that is about the normal cell division process. See, in nucleus topic, I discussed about multi-nucleated condition. One cell contains many number of nucleus. One cell contains many number of nucleus. Sir, how the multi-nucleated condition is possible? Very simple. Once the karyokinesis is completed, the cell not follows the cytokinesis. The cell not follows the cytokinesis. Once the cell not follows the cytokinesis, one cell having two nucleus. Again, if these nucleus are involved in, karyokinesis, again the two nucleus cell becomes four. Four nucleus cell becomes 8. So for that one, the best example is the coconut endosperm. We know about the liquid endosperm. Within coconut, there is an endosperm is there. That is nothing but what we are drinking, the coconut water. The coconut water is the liquid endosperm. Within that liquid endosperm, more number of nucleus are present. The liquid endosperm indicates for the nucleus only, not the cells. Nucleus only, not the cells, right? That is about the multi-nucleated condition. So how multi-nucleated condition is maintained? That's what here question, right? Here multi-nucleated condition majorly maintained by without any cytokinesis. Without any cytokinesis. Okay. See here karyokinesis is occurred. Cytokinesis is not takes place. Cytokinesis is not takes place. If the process repeatedly takes place, then many number of nucleus are formed. The condition is called as multi-nucleated condition. The condition is called as multi-nucleated condition, right? That is about the telophase. Next one we discussed about the cytokinesis process. Remember, this step is called as reverse prophase, opposite to prophase. The change is opposite to prophase, right? Coming to the last part in cell cycle, cytokinesis. The name indicates here, cytokinesis. Cyto indicates here cytoplasm. Kinesis means division. Division of cytoplasm is called as cytokinesis, right? See, within plant cell and animal cell, the cytokinesis takes place, but the process is different. Means whatever that cytokinesis takes place, okay, the plant cell cytokinesis process is different from animal cell cytokinesis process. So here cytokinesis takes place. Generally we know this is the cell, right? Here two nucleus are present. If the cytokinesis takes place, one cell divided into two cells. One cell divided into two cells because of the cytokinesis process. Otherwise, one cell contains two nucleus, that is one cell, but it contains two nucleus. If cytokinesis takes place, right? See, one cell contains the one nucleus. So one cell produces two cells. One cell produces two cells, right? So here cytokinesis, within that animal cell cytokinesis process. This method is called as cell furrow method. Within plant cells, the method is called as phragmoplast method. This one is also called cell plate method. Right? I will discuss what is the difference between the cell furrow method and the phragmoplast method. Here, very in general question is, sir, in plant cell cytokinesis, in animal cell cytokinesis, which one is difficult? Which one is difficult? Remember, the plant cell cytokinesis is difficult. The plant cell cytokinesis is difficult from animal cell cytokinesis, because guess the answer is, remember, within animal cells the cell wall is absent. The cell wall is absent. Within plant cells the cell wall is present. So here cell wall formation followed by the division of cytoplasm. So that is the difficult within plant cells. Within animal cells there is no cell wall formation. Simply plasma membrane formation is only occurred. Plasma membrane formation is only occurred. Right? So this is one of the animal cell. Here cell furrow method is takes place. That is nothing but this is the initial stage of the cell. Right? So here two nucleus are present. After the karyokinesis, two nucleus are present. So within this case, division of cytoplasm is takes place. Division of cytoplasm is takes place. All of you remember, the division of cytoplasm starts from the peripheral region. The division of cytoplasm starts from the peripheral region. So this is the one nucleus. This is the another nucleus. Here separation of cytoplasm starts from the peripheral region. Starts from the peripheral region. So finally it reaches the center of the cell. Finally it reaches the center of the cell. So here what I am telling, within animal cell, cell furrow method involved in the separation of cytoplasm. Within that case, the separation of cytoplasm starts from the peripheral region. Finally it reaches the center of the cell. So this method is called as cell furrow. So why is this called as furrow? Furrow is nothing but gap. The gap is formed from peripheral region. It finally reaches the center region. So that is about the animal cell cytokinesis process. So sometimes asking bit animal cytokinesis, plant cell cytokinesis, which one required more time? Animal cell cytokinesis, plant cell cytokinesis, which one required more time? Remember, the plant cell, the cytokinesis process required more time. The plant cell cytokinesis process required more time, because here cell wall formation and all the components here, different parameters are present here within plant cell. But within animal cell, simple, plasma membrane formation is enough. Sir, what exactly cytokinesis? Cytokinesis is nothing but majorly cytoplasm division we are saying, but within that meaning is whatever that cell organelles are there, cell organelles are divided. Division of cell organelles, that is nothing but division of cytoplasm. Division of cell organelles is nothing but division of cytoplasm, right? So that is about the cell furrow method. Other one is the phragmoplast method. For suppose this is the one of the plant cell. Here this is the nucleus. Right? Here also division of cytoplasm takes place. How the division of cytoplasm takes place here? Okay. This method is quite different. From the cell furrow method. Here division of cytoplasm starts from the center. Finally it reaches the peripheral position. How I will explain. Right here, the division of cytoplasm within the phragmoplast method starts from the center, right? Finally reaches the peripheral region. Then one cell forms the two cells. We know about the plant cell structure. This is the one plant cell, right? This is another plant cell. Between these two cells, there is a cementing substance is there. The cementing substance is called as middle lamella. Cementing substance is called as middle lamella, right? After that middle lamella, okay, the very first layer, okay, this one, the blue color line, that is indicates primary cell wall. Primary cell wall. We know the composition of the primary cell wall. For the formation of primary cell wall, cellulose fibers are required, and hemicellulose fibers are required. Pectin, suberin, all these components are required. Remember, sir, how these components are formed. Remember, whatever the components which are required for the cell wall formation, which are transported by the vesicles, especially which are formed by the Golgi complex. We know about the Golgi complex functions. We already discussed. Just all of you recollect that point. Okay, within cell division process, the Golgi complex produces some of the vesicles-like structures. The vesicles-like structures contains cellulose fibers, hemicellulose fibers. Mostly 90% of the cellulose fibers are there, and remaining percentage is hemicellulose, pectin, suberin. All these components are present. But remember, other than cellulose, remaining all are less percentage. So after that, plasma membrane formation. Once the plasma membrane formation takes place, right? See, along with the plasma membrane formation, there is a primary cell wall formation takes place. Remember, here transportation is done by vesicles. Vesicles are sensitized by Golgi complex. Right? That is about the primary cell wall formation, right? After that primary cell wall formation, middle lamella components also synthesized. See, these components also very important, which are also synthesized by the Golgi complex, which are transported by the vesicles. The components are nothing but calcium pectate and the magnesium pectate. Right? That is about the middle lamella formation and the primary cell wall formation. So we already discussed here the plant cell wall formation, or the phragmoplast method, or cytokinesis in plant cells. Started from the center of the cell, finally reaches the peripheral position. Right? So that is about the plant cell phragmoplast method. So once the cytokinesis is completed, the one cell is divided into two daughter cells. So I already said, okay, whatever that meristematic cells are there, which are again involved in the cell division process. For suppose, if we discuss about the adult heart cells which are existing in G0 phase, the finest example is the nerve cells, which are only divided once in the lifespan. The divisions are only takes place in embryonic condition, right? So whatever that nerve cells we have right now, all these nerve cells are existing in G0 phase. Some cells are not involved in the cell division, but some cells are continuously involved in the cell division. So I like to ask the question here, why plant cells are showing continuous growth, why animal cells are, all animal cells, or in animals growth is limited, in human beings, limited growth is present up to 18 years, or mostly up to 18, below 18 years, the growth is exhibited by the human beings. I said that time hormones. So hormones are there, hormones are influences the cell division. Hormones are there, growth is there. Hormones are not there, growth is not there. So metotropin is the best example for the growth hormone in humans. So in plants there is an auxins are required, cytokinins are required. Auxins are required, cytokinins are required. So in plants, whatever that plant tip pods are there, the tip pods continuously produces the growth hormones. That is nothing but the auxins and the cytokinins. So that's the reason why in plants throughout the lifespan growth is present. But in human beings the growth hormone produced up to a certain age. After that age, okay, human body not produces the growth hormone. So within humans limited growth is observed. Limited growth is observed. Means the growth is fixed for one particular age. Right? So that is about that. Some examples. So this is about the cell cycle, and see there is an interphase is there. Next one is the mitotic phase. Within that karyokinesis and the cytokinesis. End of the cytokinesis. Okay, we have here two daughter cells. Right. Next one we discuss about the checkpoints which are related to the cell cycle. Checkpoints which are related to the cell cycle. Right.